Good morning, everyone, and thank you for attending my PechaKucha, where I'll be talking about what triggers a bacteriophage. Now, what exactly are phages? Well, they're highly abundant viruses within the gut, and they have an important role in shaping bacterial communities. They exclusively infect bacteria and replicate within the host cell by using host machineries. A phage-mediated killing effect can be seen through plaque assays, which is a standard method for quantifying viral titer. Plaques are absences of bacterial growth, and these can be counted to determine the number of phages in a sample, assuming that each plaque is caused by a single phage. The dynamic between the phage and the bacteria is largely dictated by the phage's replication cycle. Once a phage injects its viral genome into the bacterium, the phage enters the lytic or lysogenic cycle. The lytic cycle results in host cell lysis and the release of phages. But during lysogeny, the phage integrates into the host genome, where it lies dormant until an external trigger causes it to awaken, lysing the host cell. Many factors influence the fate of the phage, which largely impacts bacterial communities and the therapeutic application of phages. Things like diet and antibiotics can promote entry into the lytic cycle, whereas the number of phages per cell and host abundance can encourage lysogeny. Some phages also have the ability to switch from the lysogenic to the lytic cycle. This awakening of phage when it switches from the lysogenic to the lytic state can be seen through a growth curve, where we have time on the x-axis and bacterial growth on the y. The wild-type host bacteria and the lysogen, which is a bacterium carrying a dormant phage, both have similar growth patterns. But what would happen if we were to expose it to a stressor like an antibiotic? When wild-type E. coli K12 is exposed to ciprofloxacin, a known trigger awakening phages, growth is inhibited, but there's no release of phages. HK97 lysogen is a well-studied phage model that contains a dormant phage and can undergo both the lytic and lysogenic cycle. When we expose the lysogen to cipro, there is a crash. There's a clear difference in signal here, and this is because the DNA-damaging antibiotic is activating the bacteria's DNA damage SOS response system. This causes cell lysis, which awakens the phages, causing the crash that we see in the lysogen model. We can use this drop in bacterial growth as an indicator for phages being released. A high-throughput screen has been done by a previous student using this concept to identify triggers. Height of the bar corresponds to the magnitude of drop in bacterial growth. Fluoroquinolones and beta-lactams are classes of antibiotics that are known triggers, but interestingly, other compounds were also picked up as hits. The screen lacks some controls that serves as a proof of concept. This leads us to my project, which will be focusing on investigating compounds that influence the decision of choosing to enter the lytic versus lysogenic replication cycle. I'll be identifying compounds that operate both through and independent of the SOS response system, which is a bacterial stress pathway activated by DNA damage. First, a primary high-throughput screen is done with my two models, wild-type host bacterium E. coli K12, which does not contain a phage, and the lysogen, which contains a dormant phage. The two models are exposed to bioactive compounds at a single concentration, after which growth rates are measured and hits are identified based on growth inhibitions. We carried out a high-throughput screen where each assay plate contained one bacterial strain and the addition of a plate of bioactive compounds. There are a total of 4,000 bioactive compounds to test against my four strains. Compounds that cause a crash in just the lysogen model will be operating through the bacterial SOS pathway, resulting in phage-mediated effects. Here is what the screen looks like when it is fully operational. The robotic arms move the plate between the different systems, removes the lids, and also changes the configuration of the plates to best fit each system's needs. Here the plate is inverted to dispense the bioactive compounds. We were able to identify 38 potential triggers that operate through the SOS pathway. Hits were classified as compounds that cause a greater than 50% reduction in bacterial growth in the lysogen model only. One of the highest hits was harmane, which is a heterocyclic amine found in a variety of foods like meat, tobacco smoke, and even coffee. If we were to look at the SOS mechanism, which is awakening the phages and inhibiting bacterial growth, we can see that a key player is a protein called RAC-A, which binds to the damaged DNA, triggering the SOS response, causing host cell lysis and the release of phages. By deleting the RAC-A gene, we can create a model where we test for compounds that operate independent of this response. And there is evidence that compounds can cause bacterial lysis and the release of phages independent of the SOS response. For instance, oral administration of 5 different classes of drugs was recently reported to inhibit bacterial growth, suggesting phage-mediated effects operating independent of the SOS pathway. We can explore these compounds by adding additional models to our primary screens. So in addition to wild-type K12 and HK97 lysogen, complementary models where the RECA gene is deleted will allow us to detect compounds that operate independent of the SOS pathway. Studying these can possibly lead us to new bacterial stress pathways. This is the same growth curve seen earlier with wild-type and lysogen exposed to Cipro. The crash in the lysogen model is due to the DNA-damaging antibiotic activating the SRS response, causing cell lysis and the release of phages. To explore compounds that behave independent of this pathway, we deleted the RECA gene to create two new bacterial strains and expose them to Cipro. And we do not see the same killing effect here since the SOS pathway could not be activated. Now, cells do die, and there are a few phages passively being released in the RECA mutant lysogen, but there's no increase in phages, which is why we do not see a phage-mediated effect or a crash. We can now incorporate the RECA-deleted strains into our high-throughput screen. Once the primary screen is done with all 4 bacterial strains, I will do a secondary screen with the primary hits by exposing them to the same strains with bioactive compounds being added at a range of different concentrations. I'll measure growth rate and categorize hits, but I'll also centrifuge the overnight assay, filter, and measure any increases in phage production. So why do we care about how these phages are behaving? Well, one reason is because we're consuming phages every day through our diet and the medication we take, and who knows what role they play in how they modulate bacterial populations. So the next time you have a cup of coffee, maybe the reason why you feel so awake is because some of the phages in your body have also just woken up. Thank you all for listening.